Thank you to Will and Safi for organising the event. It's like such a great initiative, and I'm just so happy to be kind of presenting my research today. So as Will said, this is my undergraduate dissertation work that I did in my third year at Durham, and it's just recently been published in Rapid Communications in Mass Spectrometry. And I'm just going to be talking to you all about it today, and it's basically how we can use nitrogen isotopes in macroalgae, otherwise known as seaweed, same thing, and how we can use them to determine sources of excess nitrogen pollution. So what's the problem? I'm just going to start by talking about the nitrogen cycle a little bit, and this is just a really simple figure that I got from the European Commission in order to demonstrate that. So we have atmospheric nitrogen, obviously N2 gas in our atmosphere, which is utilised by plants. Animals then eat these plants, use it for things like protein synthesis, etc. And then the animals decompose / produce manure, and then that nitrogen is kind of returned to the atmosphere or kind of into the soil. And you know, there are lots of other inputs and outputs and things. This is just a very simplified version to kind of demonstrate the real crux of what I want to talk about today. So then the problem comes with humans, with us. We disrupt this cycle a lot, and it's mainly through our use of fertilizers. So we put fertilizers on crops in order to produce a higher yield. And fertilizers are mainly composed of nitrogen. There's some phosphorus in there, the major nutrient, and also some micronutrients and stuff. But kind of for what we're talking about today, we're going to think of fertilizers as mainly nitrogen. They're normally in the form of nitrate, but you can get ammonium fertilizers as well. So what happens is when we put these fertilizers onto the land, they get washed away with precipitation as runoff kind of into our coastal and estuarine areas. It's kind of, I've not shown it on this diagram, but we also have sewage and manure that are large sources of nitrogen as well. That we kind of contribute to, and these also contribute along with fertilizers to excess nitrogen in the environment. So once we've then got the excess nitrogen kind of in our water bodies, then we end up with eutrophication, as I'm sure we're all familiar with. And, you know, we get microalgae growing along the surface, and it takes up all of the oxygen, and then we end up with some lovely dead fish, which is not what we want in the slightest. There's also another issue with it. So particularly in coastal areas, we get opportunistic macroalgae. So not microalgae like I was just talking about, this is macroalgae. And this is also as a result of there being too much nitrogen in the water. And it's normally green macroalgae, as you can see on the images, so you can get green, red or brown macroalgae. And they're normally annual species, which means that they thrive with a high amount of nutrients like nitrogen in the water. And this is because their growth strategy is just of surge uptake. So that means they'll take up any nutrients that are available straight away and use it directly for growth. So that means if there's a high amount of nitrogen or nutrients in the water, this species will just grow exponentially. This is kind of in comparison to perennial species that I'll get onto a little bit later, and they can actually store nutrients within their tissue, so they can take up the nitrogen from the water column, store it for a few months, and then use it for growth when conditions are a bit more favourable, so when you've got warmer temperatures or higher irradiances. So other than this green macroalgae just being a bit unsightly, it actually causes anoxia within the sediment beneath it, which is obviously bad news for anything living in the sediment, but it's also bad news for any organism that want to feed on things that live within the sediment. And this is a kind of problem, for example, at Boodle Bay, where these pictures are from, which is in Northumberland, northeast England, and you can see it's just absolutely covered in the macroalgae. And basically there are wading birds that want to feed in the worms in the sediment, and they then can't do that because one, well, the sediment's absolutely covered in this green macroalgae, so it just can't get to it, and two, even if it could, there's no worms left because they've been killed off by the lack of oxygen. So in general, it's just if you see kind of this green macroalgae growing, it's just an indicator that the ecosystem health just isn't great. So then what can we do about it? The obvious solution is to reduce the amount of nitrogen coming from the land. You know, that would be lovely if we could just do that. But we need to know what's actually causing it and what the source of the pollution is. So we need to know whether it's fertilizers, whether it's manure, whether it's sewage. And it kind of needs to be identified on more of a local scale. It's no good trying to look at it nationally or even really regionally, because the kind of outputs that are going to be affecting one area can be completely different in another area. And we can't kind of work out the source by just looking at the nitrogen concentration in an area. You know, that's just going to tell us how bad the pollution is. It's not going to give us an insight into what could be causing it. So this is where nitrogen isotopes come in, and they can give us an insight into the possible source of the pollution that's occurring. So I'm just going to give a quick explanation about the notation of delta 15N, which is how we talk about nitrogen isotopes. So delta 15N is compared to an international standard, which is air. So it's just literally N2 gas, and that is our 0 point on the kind of scale bar there. So any sample that we measure that has more 14N in it in comparison to that international standard will be negative on the delta 15N scale, and it'll be increasingly negative the more 14N that it has in it. And a kind of similar picture for 15N. If within a sample you have more 15N, then it's going to be positive on the delta 15N scale, and increasingly positive the more 15N it has in. And so this is really useful for us because each of our sources of pollution have a distinct nitrogen isotope signature, which helps us a lot. So fertilizers, both kind of ammonium and nitrate, tend to have a delta 15N value of about 0 per mil, and this is because they're manufactured straight from N2 gas from the air. So they're literally manufactured from the standard essentially. So they're going to end up being about 0 per mil. Oh, sorry, I should also just mention that per mil sign, like a funny percentage, just means out of 1,000 as opposed to out of 100, because isotopic signatures are so small, it just doesn't make sense to put them as a percentage. You've got to do it kind of out of 1,000. So if I say per mil, it kind of think of it like a percentage, but out of 1,000. And so, yeah, then we've got manure and sewage are kind of on the more positive end of the scale. And this is because life kind of tends to prefer the lighter isotopes. So life will kind of utilize 14N, and therefore any waste products, so manure and sewage, will be enriched in 15N kind of relatively to that. So how do we actually use macroalgae as our solution for monitoring nitrogen pollution? It seems a little bit counterintuitive that I've kind of been talking about, oh, this green macroalgae is such an issue and it's kind of bad for the health of the ecosystem. So what we do is we use a different species of macroalgae. So the image that you can see is of Fucus vesiculosus, and this is a perennial species, as I was kind of talking about earlier. It's a brown macroalgae, and it can store its nitrogen for kind of use when growth conditions are a bit more favourable. It assimilates nitrogen from the water column with very little fractionation. So this means that the ratio of nitrogen isotopes remains pretty much the same as it was in the water column as when it's incorporated into the tissue of the macroalgae. So that's obviously a really good thing. We could, of course, just sample the water, but actually it's quite time consuming to just do it that way. And it's also, it costs quite a lot of money. And more often than not, it doesn't actually give us a very representative idea of what's actually going on in that area, especially in kind of estuarine areas where there's just so much chemistry happening with kind of salt wedge and flocculation and kind of things like that. You could get a single delta 15N value, and that just wouldn't be representative of what's been going on for the last few weeks or so in that area, and maybe it's what pollution it's been affected by. So this is where the macroalgae comes into it. It kind of averages out the nitrogen that's bioavailable in the water column, ready for use, and it will kind of incorporate the signature of the delta 15N in that area kind of over a period of weeks. So previous work that's been done has found that translocating macroalgae is an effective procedure, which is where you'll take some tips of macroalgae from one area, put it into another area, and it will effectively kind of incorporate the delta 15N of that new area. So we know that it works, we know that's a thing. So what we want to do is just take it one step further and culture the macroalgae in isotopically-labelled solutions. So this means kind of changing their delta 15N to something really unnatural, kind of with the aim of then putting it back into the environment, and then the kind of theory is that it will converge back on the pollution point. So this is our experimental setup. So we collected macroalgae tips from Easington Colliery, which is in the northeast of England, and then set up these little cute jars with tights over the top of them to allow for atmospheric exchange. And then they were popped into an incubator, which had kind of set light and temperature settings, as you can kind of see there. And yeah, what we did is we got some artificial seawater and combined this with ammonium sulfate solution. So that was kind of our source of nitrogen, the ammonium sulfate. Excuse me. And so what we did is we aimed for a nitrogen concentration in the solutions of about 50 micromoles. That's what's consistent with the literature. Ended up about 55-ish, but not too much of a worry. And then our delta 15N was 170 for the 15N enriched solution, and then minus 60 for the 14N enriched solution. And it doesn't matter too much that these aren't kind of the same amount away from zero. All it kind of means is we just needed unnatural delta 15N values. It matters more that the concentrations were more consistent, because there is some stuff in the literature to say that concentration can affect the fractionation. Kind of, it's been observed at higher concentrations than this, but fractionation has been observed. So we thought we'd just keep it consistent with that. And yeah, we cultured for 19 days, sampled every single day, and I changed the solutions every 3 days. So this is the first of our results. So the grey line that you can see here is obviously the 170, the delta 15N of that 15N enriched solution. And as you can see, it's just a really nice linear increase towards equilibrium with that solution. So the macroalgae tips started off with the delta 15N of about 11 per mil. So you can see that the delta 15N has just drastically increased towards that signal over the 19 days and eventually reached equilibrium. There is a little bit of a tendency here. It trends away from the trend line between kind of days 8 to 10. And what we figured out is that there was some power cuts between those days. So our temperature and light settings were knocked off slightly, and we think that's why we see those deviations, and you can see it a bit more clearly later on. So this is our 14N enriched samples. Again, same, a really nice linear, in this case, decrease towards the 14N of the solution. Doesn't quite reach equilibrium like the 15N enriched solution did. And we believe this is due to the fact that this solution is not as enriched in 14N as the other solution is enriched in 15N. So if you remember, our 15N solution was about 170 per mil, and this one was only minus 60 per mil. So this one was a little bit slower to incorporate, but we still see the kind of same behaviour of that linear decrease towards equilibrium. And so these are our just control samples. Obviously you've got to do a control. And so these didn't have any of the ammonium sulfate solutions in, these were just the artificial seawater. So in theory, there shouldn't really be any nitrogen in here. So what you'd expect is for it to just stay at this background level, and which it pretty much does. It's within 2 per mil of the background kind of macroalgae delta 15N, but we do kind of see a slight kind of cyclicity between these 3-day periods. Sorry, these grey dashed lines are when I changed the solutions. So we do see a kind of slight increase until we get to here between days 8 and 10, where we had that lovely power cut, and it kind of switched us back again. And we think the kind of reason for this is that over the 3-day period, there's kind of a buildup of metabolites, for example, ammonium, and kind of subsequent reprocessing of the 14N bonds that are released, hence leaving the tissues more enriched in 15N. And we're not quite sure why it would do this, because as I kind of mentioned before, life kind of prefers the lighter isotope. So we're not really sure why it would get rid of its lighter isotope, but it's an interesting result nonetheless. And so this is just our kind of final set of results. This we did retrospectively, because with the previous graphs there was quite a significant scatter of standard deviation. We thought, let's look at the different sections of the macroalgae tip. So when we harvest the macroalgae, we literally just rip it from the kind of sample. And so what you're seeing here is we've sampled the torn base, literally where we've ripped it, the middle and the apex, which is the kind of tip of the macroalgae. And what we were expecting is different to what we got. So we were expecting for the apex to incorporate the solution the best, kind of most effectively and trend at the highest rate towards the delta 15N solution, just because it's the grown part of the macroalgae, and we thought it would be taking on the kind of most nitrogen and kind of assimilating that the best. But actually it was the torn base that's incorporated the delta 15N the best. And basically we think this is because we've literally broke it and kind of opened the tissue, and then that's kind of allowed for an easier exchange of the nitrogen, and subsequently the delta 15N has altered most dramatically at those points. We still do see a significant scatter, so this kind of suggests that the isotopic incorporation is not homogeneous throughout the macroalgae tips. So what does all this mean? I feel like I've given you a lot of graphs, a lot of data. So our kind of theory is that when we translocate these isotopically labelled macroalgae back into the natural environment, in theory, they should converge on the point of nitrogen pollution, so the delta 15N signal of whatever nitrogen pollution is occurring in that area. So yeah, both the 14N and the 15N tips should converge on that point. So for example, for case 1, if it was the chemical industry, fertilizers or nitrification, it should kind of tend towards kind of negative to 0. And for case 2, if it was sewage, manure or denitrification, which is just another process, it should be a little bit more positive. And so we set out to do this. We set out and did our, we had some field work going on the River Tees. And then, don't we all love it, Corona hit. So literally a few days after I started my fieldwork experiment, we got put into lockdown, which was just so exciting. Yeah, it was great. And so we got some nice data from the first few days. So it kind of started to suggest that it was going to kind of agree with our hypothesis that it would trend back towards the pollution point. But to be honest, we didn't get enough data to kind of say that for absolutely sure, and certainly didn't get enough data to present it. So that's something that I'll be working on throughout my master's, kind of COVID permitting. And so, yeah, still yet to be exactly proved, but we're pretty confident that that's going to be the case. So conclusions. So yeah, Fucus vesiculosus macroalgae tips will incorporate an unnatural delta 15N signal over a period of weeks. So we did 19 days, so 2-3 weeks. The torn base of the tip incorporates the delta 15N signal of the solutions most effectively. And we now kind of predict that isotopically labelled macroalgae tips could be used as a kind of low-cost alternative to monitor and also kind of determine nitrogen pollution sources. So kind of where do we go from here? So obviously I am really keen to get back out in the field and finish my fieldwork trial and kind of prove that this is definitely a thing and it definitely works. But in the meantime, I will be in the lab kind of optimizing and perfecting the methodology of how we best incorporate the delta 15N solutions and kind of run some more experiments to understand macroalgal physiology and yeah, in what way does it incorporate best. And I just have some acknowledgements just to say thank you to all the people that helped with this research, and thank you for the funding and stuff, and thank all of you for listening to me talk about my research, and I hope you've enjoyed it.
Thanks for that, Imogen. So now we'll take questions. So if people want to put them in the chat, I can read them out, or you can message me so that I can read them anonymously. Oh, we have a question already. Someone's asked, is there a reason why an ammonium solution was used as the source of nitrogen instead of a nitrate solution?
Oh, so for the for the actual experiment we used ammonium. Yeah, so there's quite a lot of literature that kind of suggests that ammonium is taken up a lot better and at a lot faster rate than nitrate is, and so we just kind of went for ammonium and didn't even really try the nitrate out beforehand. It's just kind of to do with basically ammonium can be assimilated directly into macromolecules within the macroalgae tissue, whereas nitrate has to be reduced to nitrite and then reduced to ammonium. So there's just a bit more faff involved with it essentially, and the kind of macroalgae, it's a lot kind of more favourable. It takes less energy for it to just assimilate the ammonium. So yeah, that's why we went with that.
Safi, can I ask a question? I know I always jump in, and I'm just wondering, so I was wondering if you could go back to one of the slides at the beginning where you were showing the different delta N values for different components, so for the fertilizer and things like that. I was just wondering, when I looked at it, I'll wait for you to, yeah, there you go. So I think, but when you were showing your results, there was, even in the kind of control cases, there was, as you were mentioning, a couple of mils per mil variation naturally. So I'm wondering if, with these bearing in mind that there's quite high error bars on the overlap between the different sources, is it actually significant enough to distinguish between them, do you think, in some, like in most cases? Does that make sense?
So do you mean like in the natural environment, like would these kind of values, like would you see enough of a difference to kind of yeah, so there's a kind of argument, you know, for all these values are kind of representative. Like I kind of just know from experience that like for the work that we've done that like it is kind of obvious. So like you'll kind of know if it's manure because it will just be positive and it'll be about 15 from my experience, and kind of yeah, it's more negative/0 for these kind of fertilizers. But no, it is a good point. Like there is definitely some overlap, and there's also, you could make the argument, well, what if there's fertilizers and there's sewage like affecting an area, then how on earth do you go about it? Will the macroalgae kind of incorporate that signal? And it's definitely an area for future research, and you can do something called Bayesian modeling, which kind of will kind of set out the nitrogen dynamics in an area, and that's how you go about properly distinguishing everything. But like I said, that's kind of like future research. I'm not quite there yet, but yeah, no, definitely a good point. Yeah.
Thanks.
So we have sort of a two-part question. So I'll read the first part and then. So Tamsin has asked, do you think a similar technique could be used in other ecosystems that can be affected by nitrogen pollution, e.g. coral reefs? They've said no idea if there are any analogues to a macroalgae in those ecosystems.
Yeah, definitely. So even that coral reefs, there's a lot of work done around Australia. I know a few papers from Australia where they've used different things. It's literally, I think the most important thing and why macroalgae is such a good analogue, especially with the species that we've got, is that there's barely any fractionation from the water column into the tissue. So I think if you're looking for a tool, a kind of a biomonitor to be able to use, you need to first ensure that there's no fractionation coming from the water column into that tissue, because then otherwise it's just utterly pointless. I mean, you could correct for it if it was always a known value of fractionation, but you'd obviously have to work that out. But yeah, you could definitely use anything that has a nitrogen isotope ratio, which most living things do, just as long as you either know the fractionation or there's no fractionation kind of between the environment and between the tissue.
And the second part of the question was, what challenges do you anticipate when undertaking your fieldwork besides COVID?
Oh, the cold? I don't know. No, I think for this kind of fieldwork, like off the top of my head, the most significant challenges is just like getting down to the seaweed, honestly, like without falling into the river or like falling into the sea. Like, yeah, I think that's probably the worst thing. And also just like scouting out somewhere, so when you go to translocate a piece of macroalgae into the environment, like make sure that it's going to stay there and it's just not just going to be swept away like with the tides and stuff. So yeah, I've got to come up with some like engineering controls for that, but yeah.
Olivia has asked, does it have to specifically be that certain species of macroalgae?
No, so it doesn't have to be. And the reason why we choose this species is because, well, firstly, it's kind of been done by the literature, so we know a lot about it already. Also, it's really kind of popular throughout the UK and Europe, so it would be really easy to kind of replicate, say if kind of environmental agencies wanted to use it. But no, it doesn't have to be this species. We know with this species that there's little fractionation, which is why it's so kind of good to use. But you could use a different species, definitely, just again, as long as you constrain the fractionation and make sure there's not too much going on, you could definitely use different species, yeah.
Okay, so I think we're going to have to move on just to make sure we keep to time because of how many questions there have been, which has been fantastic. So thank you very much, Imogen, for doing that. It was really interesting. And now we'll move on to our second talk, which is from James Mulqueeney, who is at the University of Bristol doing in his 4th year of his integrated masters. So presenting work which was done over the summer as an independent research project, and is now continuing with the work as part of his masters. So James, if you want to go ahead.
Yeah, no problem. You see that?
Yeah, there we go.
Is that all good?
Yep.
All right. Thank you, Will. So hello, everyone. Welcome to this talk. So hope you're all well. I'm James, and today I'm going to talk to you about the impact of fossil calibrations on constraining the evolutionary timescale of green plants. So to walk you through, I'm going to spend about 15 to 20 minutes talking about some research that I performed over summer. And if you have any questions, feel free to ask me at the end or drop me an email at the email on the screen. And yeah, I'll put that in the chat afterwards. As I'm flicking through the slides, I'm going to pause for around a few seconds just to allow you to take it in. So yeah, let's start. Okay, so to outline the talk, I'm mainly going to be discussing about green plants and the fossil record. So the fossil record is quite disparate, so it's very wide, and I'm going to talk about how it varies through time, how it changes from recent species to very ancient species. I'm going to highlight how that influences current molecular clock analyses and how it relates to the times that we're going to estimate. And the most exciting bit, I think, is the projection of a new accurate time frame for the evolution of green plants. So why are green plants important? Well, for one, they're fundamental in controlling life processes. So for instance, they provide all the needs to the trophic system, so providing food for herbivores, which is obviously top down. And then they played a role in the colonization of land in freshwater environments. So for instance, Cooksonia over here was, this is a model of that, which was one of the first land plants, and that would have allowed for animals to colonize land, which is really quite interesting. And also, which is one of the key factors that we still see today, is the controls of atmospheric compositions. So for instance, providing oxygen and taking CO2 out of the atmosphere. Okay, to introduce the premise of the talk, I'm going to talk a little bit about molecular clocks, as that is mainly what our analysis focuses on. So to basically sum it up, originally, our estimates for how old different species would come directly from the fossil, from the rock record and the fossil record. So for instance, we would find the fossil and we would date the age of the rock that it's found in, and we would use this as the time that they would have originated. But there's problems with this. So for instance, the fossil only provides the minimum age of how old these species could be. It could go further back in time. So this is where Zuckerland and Pauling invented something called the molecular clock. So the use of the molecular clock was quite difficult to use up until the advancement of computers, where it's really become revolutionized within the field. So to explain how this works, is they essentially use genetic data with fossil calibrations in order to constrain the age of species and different groups of organisms. But the problem is with these estimates that we do from this is that they're heavily reliant on fossils, on the selection of fossils, and thus the plant record, the estimates we have obtained for plants at the moment seem to be inconsistent. So why is this the case? Well, as I just highlighted, the quality of the fossil calibrations and how good we understand the fossil record is one of the key contributing factors as to how old we estimate the species to be. So for instance, trilete spores and tetraporina, two classically used calibrations within the analysis of green plants. However, they have since been replaced by different calibrations, as these ones are seen as more obsolete and they're too old for what we have for current estimates. So how do we overcome using obsolete fossils? Well, in 2012, Parham et al. Produced a criterion on how we should select fossil calibrations. This is basically composed of 5 things, but we can mainly break it down into 3. So the first part is when we're suggesting a fossil, we should always use the museum accession point. We should always reference it to which museum number it's from, so its accession number. We should justify its age, so we should point out as to how it's been dated, what formation and stratigraphy that it's found in, and how it relates. And then the third bit is its phylogenetic justification, so how it relates to different organisms that are also found within the tree. But the problem is, not just with molecular clock analyses of plants, but in general, is that studies have chosen to ignore this criterion. So for instance, the 3 papers that are showing you, are highlighted in this one here, these are all 3 examples of where people have produced molecular clocks for plants, but have failed to fulfill the criterion proposed by Parham. And now I'm going to show you how that has basically affected data and estimates and why it's a huge issue. So the first thing we need to know is, how did we analyze that? So this year, there was a paper released by Ni et al. That focused on the evolution of plants, green plants in particular. So we're going to reanalyze this data set in order so then we got a controlled version, and then we allow for a direct comparison between 2 different age estimates. So to tell you what data this made up, it was essentially 81 chloroplast protein coding genes, which were for a total of 97 green plants and 2 red algal species. So the 97 green plants were used between a variety of different green plant clades and different groups of green plants. And the 2 red algal species are basically used as an outgroup for these. So to explain that is something a bit more distant related from the 2 that allows us to know once they diverged. And when we had this sequence data, we found that there was problems with the data. So there was saturation in it. which leads to incorrect dating estimates and they're often found to be too old. So in this case, we partitioned it into 3 different sections, and we removed the third codon partition, but I'll talk on that later. Okay, so I've elaborated on the molecular data that we've used, but now to elaborate on the fossil calibrations. So basically, we have a total of 21 fossil calibrations that range across different species of green plants and different groups of green plants. And we selected these all under the criterion of pattern. So for instance, here are the 2, is the oldest and the youngest of the fossils that we used. So Bangiomorpha pubescens was published in a paper by Gibson that gave an age of 1.03 billion years old. Whereas if we look at Paleoclusia, which was by Barbara Montoya, the age of this fossil is only 86.5 million years old. So it's considerably different between the 2. And the key difference is between the organisms. So there's a long way of evolutionary timescale between the 2. Okay, so now applying these fossil calibrations, you can apply them either as soft or hard bounds. Soft bounds is what we opted to do. It allows for a little bit of discrepancy between the values and allows a bit of flexibility for values outside of the fossil calibrations, which is often more useful in these kind of analyses. Okay, so this might be a bit complex terminology for some people, but I'll walk everyone through it. So for our analysis, we performed it in a software called MCMC Tree, which is within PAML, which is a phylogenetic-based software. And as I mentioned earlier, we used the first and second codon partitions in our analysis, as we removed the third one due to the saturation. And the really cool thing is about partitioning the data is that we're able to use more clocks, which in turn makes a more precise estimate. And then this is the more complex bit where we used a general time reversible model with a gamma 4 rate. So to walk you through that is basically a general time reversible model allows to monitor the difference in the numbers of base compositions. So for instance, we've got here like cytosine and guanine, etc. So it allows to, they're not necessarily going to be equal throughout the genetic data. So that accounts for that. And then the gamma rate over here, that accounts for these different rates between the transitions and transversions. So for instance, transitions are more likely to occur than transversions. So the use of that input parameter allows us to monitor that. So once we selected the model and we've got the partition data and the fossil calibrations, we're ready to run the analysis. So we ran this using an approximate likelihood method, which basically means that it simplifies the data down so that it's not too computationally demanding and it doesn't take too long a time to run. And we use this using independent rates. So independent rates basically means that the rate of change between the genetic codes, so between say C to T, and how that's changed through time, is independently calculated through the actual genetic data that we used itself. Okay, so now on to the results. So this figure here on the right was our main figure from our investigation. So this here is a time tree of the 99 species of plants, ranging from the oldest at the top to the youngest at the bottom. So for instance, we got the age of green plants, which is viridiplantae, which is just here, around here, which, as you can see with the date, is really quite old. So 934.6 to 1204.6 million years ago. So that dates the origin of green plants to around 1 billion years ago, which is really quite scary. And to embryophyta, which is land plants. So we're looking at a large gap between the origin of green plants themselves and once they transitioned into land. So this occurred between 468.4 to 498.2 million years ago. And then finally, the bottom one, which is one of the key ones as well, is the origins of angiosperms. So angiosperms are flowering plants, which are very common in our terrestrial environments today. And these only came around at 172.2 to 233.5 million years ago. And then just to give you a brief comparison between these two, between our results to NEES, you can see that there's a huge discrepancy between how much difference there is between the older end of the tree and the younger end of the tree. So for instance, for green plants, we get an origin date on our estimates that are 220.3 million years younger, whereas for embryophyta, it's only a difference of 6.9 million years. And I'll elaborate on that soon. So basically, how did change in the fossil calibrations affect our results? So this figure here in the top right is a comparison between our results and those of NEE. So once we plotted the line across the middle here, across the center, is an x equals y line. And if all the values were to be equal to each other between the two estimates, we'd expect them all to lie exactly on that line. So for that matter, the R-square value would be 1, as there's no deviation between the results. Whereas in our results, we see there's a difference of 0.937. So there's quite a bit of a difference between the results, though. But really, it's not that huge of a difference. But as you can see, that the greatest degree of change between these nodes all comes towards the higher end values. If you look towards the bottom end of the tree, there's really, the confidence intervals are really small, which are these lines going across here. And you can see that they lie exactly within the tree, along the line, sorry, which is really quite interesting. Whereas once we start to move up, you can see that this progressively moves away. So for instance, this here is our oldest age node. So that there is here for this one here, which is Viridiplantae, and it goes back quite a while. And as you can see, it's really quite different from what we'd expect. But how do we find that these fossil calibrations, how do we find the estimates that we produce? How did they fit within the ranges of the specified fossil calibrations? Well, we found that they actually fitted pretty well. We only found that there was one group of organisms, the Charophyceae, and their fossil calibration. That was the only clade that we had that produced estimates that fell outside the calibration range, which is pretty interesting. I'll explain why that is later, but for now, I'll walk you through the bottom figure. So this bottom figure here is basically monitoring the precision of our estimates. So believe it or not, the values for NEE were slightly more precise than ours. So for instance, the R-squared value is slightly better than ours, which indicates they're more precise. But the more interesting thing is the line equation. The line equation allows us to measure the uncertainty, the average uncertainty change between a million years. So if every million years added, we get an error of 0.345 million years added to our estimates for NEE and 0.357 for our own. So as you can tell, over time that progressively builds up, and that can explain why the precision of the later estimates aren't as good as the younger ones. Okay, so what do these results mean? How significant are they? So where we changed our fossil calibrations the most, which tended to move towards the back end of the tree, they had the most change. So if I show you these fossil calibrations here on this tree, they're indicated with these little 2 numbers here. So for instance, for green plants, we use the fossil calibration from 940.4 to 1891. Whereas NEE was using it, NEE in their estimates did not use an upper bound. They simply used a lower bound for the last 6 nodes. So from embryoworth and embryophyte onwards, so from there onwards, they didn't use an upper constraint, which in turn can lead to values that are way too high. So as Warnock suggests here, an introduction of a new upper bound will have we agree that the introducing a new upper bound will help to minimize the actual capability of the estimates to go too far back in time. So in turn, that makes our estimates more accurate. And the interesting thing is how we've changed these fossil calibrations, and now that the results using the exact same genetic data, etc., are more in line with previous studies. So for instance, Morris is one of the most published recent estimates for plant divergence times. We found that our estimates fit very much within that. But as I said earlier, TETHYS didn't obtain an age that was within their fossil calibrations. And McCall had pointed this out in 2004 that the fossil record for TETHYS was not great. So it's been 16 years since then, and we've still not improved it. And the scary thing is that us and NI both found that our fossil calibrations, they weren't adequate enough to make sure that the divergence time estimated fit within that range. So what can we conclude from all of this? Well, for one, we have established a new accurate time frame for the evolution of green plants. So today, once I get this published, it will be the latest time frame for green plants. So it'll be the latest time tree today. We found that green plants evolved in the late Mesoproterozoic to the early Tonian. So in layman's terms, that was around 1 billion years ago. So quite way back in time. But there was quite a big difference between once they started to colonize land. So this was only in the late Cambrian and the early Ordovician, which is later than 540 million years ago. So it was around 500 million years ago. And the really interesting thing is that once plants started to colonize land, this is when we start to see a really big proliferation of life. And then, of course, a few million years later, we eventually get life on land, which would have taken advantage of these embryophytes. But ultimately, in our analysis, it's still limited because there's incomplete knowledge, especially within the ancient plant record and within TETHYS. So that's why these estimates seem to range so differently between everyone. It's purely the fact that the ancient fossil record of plants is not great, and also the fact that people continue to argue over the different which groups of organisms certain species of plants belong to, which of course affects the molecular clock analyses. And the final thing that we can take away from our estimates is that we could improve the precision of it by adding a greater number of clocks. So that will be done through using more partitions. But the thing is, there's no point improving the precision unless the actual estimates themselves are accurate. Therefore, we haven't done that at present. But I'd just like to finish with some acknowledgements. So I'd like to thank Professor Philip Donahue, Davide Pisani, James Clark, and Emma Landon, who all helped me with my analysis and helped with me to write it up and depict the results. And I'd also like to thank Will and Safi for allowing me to present this talk to you. Here are the references. If you want any of the references, feel free to ask me to send them over to you. But for now, any questions? Thank you.
Thanks for that, James. So yeah, same as before, just put your questions in the chat.
I guess I'll start if no one else is going to ask one with my inevitable questions that I always have. So yeah, thank you for doing that. It's really, really interesting to see. I don't really usually, I've tried to kind of avidly avoid paleo stuff for the last 3 years, so apologies if these are quite simple questions. But when you were doing your kind of the simulations to, when you were going back slightly to the simulation side of it, the results that come from that, are they, yeah, so is that a unique outcome, or is it kind of an averaging of a number of an ensemble of results? Or
okay, so basically it's an outcome from 2 different results. So where I said it's partitioned into 2 different sections of genetic data, it allows it to run 2 separate clocks. So it runs the analysis basically twice using the 2 different sets of genetic data. So the first codon and the second codon will not contain the same genetic data. Instead, they'll be different. So once you apply the fossil calibrations to it with the different rates of mutation, you'll get slightly different ages. So basically this plot here is emergence of the averaging between them 2.
okay, cool.
And the more we partition it, the more precise the estimates become, because the average starts to basically compile on top of each other.
Okay, brilliant. Thank you.
No problem.
We have a question from Harry, who asks, do you think your estimates for terrestrialization have any bearing on the terrestrialization of animals?
I think so, because I mean, there's obviously quite a delay between the two, but yeah, I mean, they have to have fed on something, and the fact that all the literature refers to the origins of embryophytes, which also coincides with the Cambrian. So we're looking at where life has really started to, animals have really started to develop how they, their morphologies, etc. Even if that is within the oceans, we still see that we've got these accelerated rates of evolution going on at this point in time. So I think, yeah, there's definitely links between the two. The links, how much one's evolving with the other will allow more opportunities for different organisms to exploit different niches and different environments. So I think that really contributed to how these animals were eventually able to transition onto land.
Okay, so I will keep checking the chat for the next minute or two just to see if any more questions come up. In the meantime, you're forced to listen to me speak again, I'm afraid. But thank you very much, James, for doing that. It's a really interesting topic. And I'm just going to also put a feedback form. It's the same link as earlier, but just in case you didn't grab it at the beginning, if you do want to leave some feedback either for myself and Safi about the talks in general or for either of our speakers, and we can pass that along to them as well. So we've got 2 more talks coming up this term. We've got one next week, which is focusing on meteorites, and then one the week after that, which is seismology-based stuff. So if you fancy coming along to either of those, have a look out for our adverts on Facebook and Twitter and any other social media that we can try and get you on. Other than that, thank you very much for coming along again. It's been a really good turnout again, and hopefully we'll get to see everyone again in the next few weeks. Yeah, it's really brilliant to have a lot of people engaging and a lot of people coming along to support, and it also reminds me to tidy my room at least once a week. So thank you for that. And if you are interested in giving a talk yourself at some point, then do get in contact with us either through our website, which I'll also pop in the chat, or you can find us on Twitter or Facebook and just get in contact with one of us one way or another. If that's everything done from me, I won't keep you any longer, but I'll keep the Zoom going for a little bit just so that you guys have got access to that form in the chat if you do want to leave some feedback, and I'll also drop a link to our website as well. We've also got a YouTube up and running now, so if you want to catch up on any of our previous talks as well, then you can find that through our website as well. But thanks very much for coming, and hopefully we'll see you all next week as well.